Good afternoon, everyone. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research. I'm on the California campus up in the executive suite. We have about 1,000 scientists busily working away despite the restrictions imposed by the pandemic. We've got all kinds of safety and testing measures in place, and we're trying to do research as best we can. So welcome to the Front Row. This is our periodic lecture series where we feature scientists from Scripps, and we try to bring you the latest, greatest developments. We've had quite a series on COVID-19, and we're trying to mix it up a little bit. So today, it's the other pandemic. So there's a real epidemic of epic proportions concerning the use and abuse of opioids. So opioids are tremendous in pain management, but they are also a scourge in that they're the most addictive substances known to mankind. And so we have two of our star faculty members here that are going to be talking about ways to change the way we do opioid medications and to treat addictions. And there are two distinct approaches. So first, the speaker we'll start with is Professor Laura Bohn. And Laura is a professor from Scripps, Florida campus. She's a unique blend of a biochemist and a neuroscientist. She studies, opioids and GPCRs and tries to find new medications. So Laura got her PhD in biochemistry at St. Louis University, and then she did postdoctoral work at Duke with Mark Caron and Robert Lefkowitz. And she started her academic career at the Ohio State University. We recruited her to Scripps, Florida in 2009, where she's been ever since. So Laura is a real hardcore biochemist, and I'm sure that you're gonna enjoy her presentation. That's gonna be followed by Professor Kim Janda. So Kim is a member of the chemistry department here at Scripps in La Jolla. He got his PhD in organic chemistry and at the University of Arizona. And he's evolved slowly over the years to start working on antibodies and the treatment of addiction. So he started as a postdoc here at Scripps in the laboratory of Richard Lerner, who was the president for 25 years here and really made Scripps a named institution. Kim joined the faculty in 1989 as an assistant professor, and he rose through the ranks where he's now a full professor. He's got a range of research interests. He's worked on catalytic antibodies, he's worked on organic synthetic methods, and most recently, he's working on developing antibodies against cocaine and nicotine and other substances. And this is a way to treat the addiction by binding to the very compounds that are circulating in your blood. So we have two really different approaches from two different campuses, and I really hope you enjoy the presentation. So again, welcome to the Front Row and enjoy the show. So Laura, take it away.
Okay, so thank you all for joining us today as we walk you through a little bit of how we approach this very important problem of addiction and our research that's ongoing here at Scripps. As Jamie mentioned, I'm coming out of Scripps, Florida, and that's just a little picture of it there. Okay, so whoa, why is it not moving? Okay, there we go. Okay, so I'm going to give you a little overview of our approach, and I'm going to start with a brief history of opioids, drugs, and how did we get where we are. And by that, I mean this opiate epidemic. I'm going to talk about opioids and pain and side effect profiles that are associated with treating pain. I'll define what opioid receptors are and describe how opioids cause cells to respond. And then I will talk about our approach to fine-tuning receptor signaling by using a combination of biochemistry and pharmacology and drug discovery. If you have remaining questions about addiction, about treatment, about therapies, I encourage you to look at the NIH National Institutes on Drug Abuse website here. It's www.drugabuse.gov, and that contains an enormous amount of resources about addiction. So how did we get here? There are essentially, has been described 3 waves in the opiate overdose deaths crisis of this epidemic. And this is shown here from a graph that's provided by the CDC and the vital statistics. This is deaths per 100,000 people in population. And they describe, and I'll break it down into the waves here, in 3 waves. The first started in the late 90s. And many of us will remember this, that there was an increase in prescribing of opioids for pain. You will recognize common opioids as OxyContin, Percocet, Vicodin. And this correlated with a time where pain was identified by medical professionals as a vital factor, such that if you had pain, your medical provider was told that you shouldn't have pain and you should be given something to alleviate pain. And with that, we saw an aggressive marketing campaign to physicians to prescribe. By the manufacturers, there were outright lies about the addiction potential of these therapeutics. And by the increase over-prescribing of these medications, we had a market that was flooded with pills. So if you think about it, you go to the dentist, you have a root canal, you may come out with 40 OxyContin tablets. You may not take all of them if you're using them very responsibly, but they sit in the medicine cabinet and eventually somebody begins to take them. So that was the first rise, was the prescribed opioids flooding the market. This then transitioned to the emergence of heroin. And so this is again deaths. This isn't use, this is deaths resulting from. Heroin started to come back on the market around 2010. I don't mean market, I mean the streets, because heroin is a substance one abused substance that is illegal. Heroin became cheaper and cleaner for manufacturing. So no longer did you have the dirty tar heroin that you have to heat up in a spoon and inject into a vein. Heroin became very clean, very potent, and could be snorted. And so this really encouraged a spike in heroin use. It also happened at a time when prescription monitoring came about. And with prescription monitoring, it became harder to get the opiates filled, the prescription opioids filled. So you had patients that were not being given a substitution. They just simply cannot have more. And at that point, there becomes a dependence and a reliance on having the drug, physical as well as psychological, as well as a substance abuse disorder that develops. And so heroin began to rise, and overdose due to heroin began to rise. The third wave came with the appearance of fentanyl. And fentanyl are a class of chemical compounds. Fentanyl is also a clinical compound. It's used for the alleviation of acute pain. It can be very useful because it has a very quick duration of action. It's cleared very quickly. It's used in surgery. But fentanyl and its derivatives, some of them are just extremely potent. And if they show up in a counterfeit pill, which we're also beginning to flood the market, you may not know just how much fentanyl is in that pill. And so since it's so potent and so strong, a little bit of fentanyl could very much surprise an individual thinking that they're taking a prescribed opioid with a bit of fentanyl in it, leading to the spike in overdose that we began to see around 2014 and continues to date. So this is some of the biggest issues that underlie where we have become. Prescribed opioids, fentanyl still remain very useful therapeutic compounds, but there was a problem and we're reaping the effects of that problem now. So I'm going to introduce just basically the opioids. It refers to compounds that are from the opium poppy originally, and these were the poppy. You can get opium from this latex that's secreted or that drips from the plant. It can be dried. And in the early 1800s, morphine was the first natural product isolated, and it was taken from the opium poppies. This revolutionized the ability to treat pain, particularly in times of war, in surgeries and so forth. And And it was also the birth of pharmaceutical manufacturing. So it's a fantastic molecule. It's wonderful. It is so helpful if you've ever been into into surgery or an accident with severe pain. You know that the opioids are very effective. Opioids act by binding to the mu-opioid receptor, and that's where we come in as these biochemical mechanics. So the opioid receptors are located throughout the body. They are found in the brain, all throughout the brain in different neurons. They are found in the spinal cord. They are found in the periphery as well as in the colon, in the small intestines as well. It's an interesting array of distribution because the activation, say, in the colon will have different effects than the activation of the receptor in the brain. And I think one of the best examples of this is a very common drug that is sold over the counter called loperamide, which is also known as Imodium. So Imodium is an opiate. It binds to the mu-opioid receptor. It's very potent, but it doesn't enter the brain, and it's restricted in the periphery, and it's used as Imodium to treat diarrhea. So it will bind to the receptors in the colon, in the neurons in the colon, and stop secretion and diarrhea. So this is an example of a drug that is an opioid but doesn't cause a lot of addiction potential because it doesn't get to the brain. Okay. You also note that it doesn't cause respiratory suppression, as that is also modulated by the brain. So to treat pain, we seem to need to get to the brain, at least for opioid receptor activity, but getting to the brain also means a risk of respiratory suppression. So what are opioid receptors? Receptors reside on the surface of neurons, and this is a picture of a neuron that is labeled this is actually a serotonin receptor, but it's the same sort of thing where you have receptors on the surface and the receptor then binds to a drug, and the drug, this is the outside of the cell, causes a change in the shape of the receptor, and that change on the inside changes the way certain intracellular proteins bind to that receptor. And in this way, the receptor acts as a conduit of transmitting what the drug has to say to the cell to mediate a biological effect. And the interaction with the proteins inside the cell will determine what the receptor has to say and what the drug has to say. So what do opioids do? When I talk about agonists, which I will, agonists turn on opioid receptors in the neurons. So what are opioids? Here are the ones that we take: morphine, oxycodone, hydrocodone, fentanyl, heroin, methadone, buprenorphine. These are the ones the body makes: the enkephalins, the endorphins, the dynorphins. They all bind to the receptor and transmit a signal inside the cell. And when this happens, we get a whole host of effects. We get pain relief. Upon continued treatment, this pain relief starts to go away and we develop tolerance. We can be rewarded. It makes you feel good. It can cause physical dependence, reliance, addiction upon chronic use. It will be used to suppress a cough reflex. Many of you may have used it for that in the past. It will suppress diarrhea, and this after long-term can lead to constipation. And then finally, one of the most severe effect of the opioids, very acute effect, even with a single dose, can be overdose, respiratory suppression. And this is preventing breathing, essentially. So the Bohn lab at the Scripps Research Institute, our goals are in this receptor is we've asked, can we activate the mu-opioid receptor, this conduit, in a way that'll turn on effects we want, like pain relief, and avoid side effects, like some of the ones I just mentioned. And in our research, we hope to discover how opioids will cause these effects at the cellular level. Exactly what are those proteins I showed you inside the cell? And once we understand this, can we figure out if some of them cause side effects or some of them are better for providing pain relief? And also in our process, we hope to understand new ways that we might go about developing therapeutics that would treat ongoing opioid dependence, overdose, and addiction. So how do we do this? So opioid drugs will turn on and off receptors. And what do I mean by that? So this is a schematic showing a receptor, and you'll see this throughout my talk. This is the cell membrane. This is outside. This is inside. The drug travels through the blood, comes in contact with the outside of the neuron. The agonist binds, activates what we call a G protein. These bind, and the G protein goes and tells the cell what to do. So this is on state. The antagonist will do the opposite. It will come in and take an on receptor and turn it off. And this is very basic terminology, but it will really help walk through what we're hoping to try to do. This I show as an on-off switch because that's how we used to think about this entire class of G protein coupled receptors. Agonists turn them on, antagonists turn them off. So here's an example of what this looks like in the lab, an example of an agonist and an antagonist in cells. We start with cells that express the receptor or a piece of brain. We break the cells open and centrifuge to bring down those membranes that contain the receptor. And then we incubate those membranes plus the drugs we want to test and put in some radioactive GTP. This is a molecule that binds to the G protein. And as the levels increase in response to drug, we see that the receptor is activated. So here's fentanyl as the drug, and this is increasing with the increase of fentanyl, the amount of drug, the activation of the G protein. So the receptor here is on switch. An antagonist, here we're putting naloxone, which is also an opiate. It's an antagonist, Narcan. If you put that on in the presence of fentanyl, it turns it off in a dose-dependent manner. So this is the simplest concept of an agonist and an antagonist, on and off. If we look at that in a mouse, as an example, it recapitulates. So I'm going to show you data on respiration in a mouse. So we use pulse oximetry, and you may be familiar with this. This is a little clip on the end of your finger that can tell you your oxygen saturation. This is very important. You want to stay around 92 to 100 in saturation, your arterial O2. It's pulse oximetry, and it recapitulates in a mouse such that if you give a mouse fentanyl, there he is steady at 99, doing quite well. This is quite a few mice. You give them fentanyl, and we see a drop in arterial levels of saturation. This is a dose of 1 milligram. It's not an extremely high dose, but it's enough to really drop respiration in the mouse. Okay, so that's an on. This is what happens when you give the mouse Narcan. 15 minutes later, you can reverse the fentanyl-induced effect. Now you see it doesn't happen immediately, and that's one of the problems ongoing about Narcan or naloxone being able to rescue fentanyl effectively, because fentanyl is a very, very strong agonist, and some of the derivatives of fentanyl are even much, much more potent than fentanyl. So this is on and off. So we have asked in our lab, agonists turn everything on at once. Is there a better way? Can we develop compounds that maybe, instead of turning it on and off, maybe gives us an opportunity to turn on part of an effect like pain relief and not turn on the respiratory suppression? And we think about this like a ceiling fan. If it's hooked up to an on-off, you get all the lights on or all the lights off, and all the fan on or all the fan off. If we had a nice dimmer switch installed, we could have on dim lights and on full fan, for example. So this is how we're thinking about these receptors, rather than single switch conduits, but more as a more complex model. So I've told you already that this G protein is important for turning on the receptor, but life is more complicated than that, because there's a lot going on inside of the cell. And this is showing a couple different G protein coupled receptors and all the different proteins that they engage with. And these are other types of receptors that are there. Here's that membrane again. look at all of the things that we can interact with. So it's possible that we could fine-tune responses and find ways to make receptors signal maybe a little differently than just through the G protein. So this could be an opportunity. So can we separate these side effects, the ones in red here, from the things we want to keep, in particular pain relief? And so in a sense, in the simplest sense, instead of having morphine, which does all of these gears, all of those different potential signaling pathways that could be downstream, maybe we could take out a couple and refine it to where we just keep what's associated with pain relief. And over the last 100 years of this research, and it has been 100 years of this research in developing compounds, it is pretty clear that we want to maintain that G protein signaling. We want the G protein to come in because drugs that do that are shown to have analgesic effects. So rather than a binary switch, we see these receptors as microcircuit integrators. We began looking for other proteins, and the way we figured this out was years ago, about 20 years ago, we made a mouse that didn't have a certain protein called beta-arrestin 2, and this mouse looked better with morphine. It had fewer side effects, and it still had some, but it had fewer side effects than we saw in a normal mouse, and yet it still had all the pain relief. So we set out trying to make drugs that would not activate this beta-arrestin component because they are both proteins that bind. And we said, what if we could only keep this on and avoid this interaction where beta-arrestins may actually lead to other signals that could come into play? Most opioid agonists seem to be good at turning on both morphine, fentanyl, what I've shown you. So this was really our challenge. So to do this, I am summarizing here 10 years of research in one simple slide that this is led, the chemistry here led by my colleague up in the corner, Tom Bannister, to really discover new molecules that could, and modify these to see if we could have potency in, say, that G protein assay and avoid the beta-arrestin interactions. And that's what we've done. Here's the compound 1718. It looks very similar to morphine and fentanyl here in a G protein assay. I've shown in Keflin for reference. And here's the arrestin assay, the same types of curves I explained. Here's fentanyl, very potent. Morphine, less, it doesn't give as high of an effect, but it's still potent, and that's what matters. SR 1718 has very little potency and efficacy for recruiting beta-arrestin. So we took this compound forward, we described it, we've published this work for those of you interested scientifically to look at the details, a great many details, and compared it to a number of other compounds. And we described what we call bias factor, which is essentially a mathematical analysis to determine the preference for G protein signaling over arrestin. We determined that it worked in animals and brain. We determined that it was selective for the mu-opioid receptor. We made sure it didn't do anything if the receptor wasn't there, suggesting it's selective. And we made sure it got into the brain. And then we tested it for its ability to relieve a pain response in a mouse model. So this is a hot plate test, which is like putting your hand on a hot sidewalk. It takes a second, maybe 5 seconds, and you pull it away and say it's hot. That's pretty much what the mouse does. And we can look at each dose and see how much it's able to alleviate the sensation. Here's fentanyl, it's very potent. Potency is this way. Morphine is also potent, and 1718 is as potent as morphine. When we do that respiratory test, which I showed you a little bit ago, here's fentanyl, very potent at inducing this very low doses. This is 1 mg per kg because it's logarithmic. Here's morphine at 10 mg per kg. Here's 1718, up to 48 mg per kg. We still see no effects on respiration. So it's very encouraging to suggest that maybe we can separate those two pathways and get rid of some of that overdose potential. We then compared a therapeutic index for a whole bunch of compounds in the set, and we saw that if we improve the separation of G protein signaling over beta-arrestin recruitment, we could improve the therapeutic window, which means you can get more antinociception before you're going to see the respiratory side effect. We also showed that fentanyl had a liability, that you're likely to see the respiratory events before you see the pain relief, or at least at the same time, so a very narrow window. We've gone on to test this compound and show that it has efficacy and potency in a mouse model for chemotherapeutic neuropathic pain. This is like a Bohn filament that tests sensitivity to touch after chemotherapy treatment in the mouse. And so this is encouraging. And we've also investigated whether or not it would, after chronic use, cause tolerance. So tolerance is the need to increase doses to maintain a therapeutic effect. And so here's a hot plate test that I showed you before, like the mouse paw on the plate. And we measure how long it takes to remove the paw when we just give saline every day for a week. So they give just nothing, a little salt water, real low concentration of saline, and they respond to morphine. And then after 7 days of morphine, we see a twofold shift in the potency. So it takes twice as much to get to where you need to be, or at least twice of the ED50, which means it actually takes a lot more to get to the maximum effect of the drug. We did this also with oxycodone. This we gave orally every day. And here's the amount of oxycodone you normally need. And here's how much you need if you've had an oxycodone every day for 7 days. So a threefold shift in potency. And the mouse too, if we did 1718, the same dose as oxycodone every day for 7 days, there was no change in the potency. So that means that the drug still acted the same on day 1 as it does on day 7, which is, I think, a very good thing, because if the drug doesn't start to wear off, you might have to take more to alleviate the pain. So we think that is an advance in developing these compounds. It's not a miracle compound, because after you have chronic use and you stop taking a drug, and you try to go off of it, there's signs of withdrawal, and this is called physical dependence. Physical dependence is overcome by weaning somebody off, so you go lower and lower doses. Here in the mouse, we took those mice that had the chronic treatment, and we took away the drug, and then we measured their signs of withdrawal. And in a mouse, it's tremors and jumping. And we can see here that here's morphine. It's pretty dramatic. It's the same scale, and it lasts for about 3 days. That's the extent of what we measured. For the 1718, they still show some signs. So there still is some withdrawal signs, but it seemed to go away a little more quickly and didn't seem to be as severe. And this could be because the compound wears off a lot slower than morphine does in the mouse. So the big question everyone always wants to know is, will it be addictive? I will tell you that we don't know that answer, because mouse models are very limited for asking, is something addictive? The standard to do experimentally is an IV self-administration, but we've tried, we cannot do this. Others have tried because of solubility problems. We can't get it in solution enough to put it into an IV. This could be a deterrent if you're afraid of abuse, but it is an issue with the compound. We do see a little bit of condition place preference with the compound, so that suggests that the animals don't hate it. They would probably associate, they do associate a place where they've had the compound before where they want to go again, but it's not indicative of addiction. It's just an indicator that it's reinforcing. And then I would leave it with saying that just treat every new opioid compound, no matter who develops it, as though it will have an abuse potential, and to handle it carefully and to be very careful in prescribing. And so that would be my take home, and this one included. So our lab continues to do the biochemical exploration, whether this is really beta-arrestin or if this is something else in all those neurons, and which neurons are really acting to regulate all of these physiologies. And in addition to the opioid receptors that I've just shared with you, to know that our lab works also on other approaches for treating pain, including kappa opioid receptors that you've heard about recently in this series, ways to eliminate some of the side effect profiles associated with those, such as sedation and diuresis. We also look at cannabinoid receptors. We have very active programs on cannabinoid receptors, And finally, we also look at other receptors that I won't have time to talk about today, but we apply very similar approaches to investigating these to improve therapeutics without side effects. I'd like to quickly introduce my lab. This is the group that really worked on the beginning of what I've said. Most of these people have grown up and moved on, have gone on to jobs in industry and in universities where they're doing their own research. Colin Schmidt was one of the pioneers on the project, as well as Kim Lovell and Nicolette Ross, who were chemists in the program, including Nicole Kennedy, who's our current chemist working with Tom Bannister. We have a few others here that are still actively working in the lab, Ed Stahl and Agnes and Savita Kanabal and Fani Pantuli. And they're here in the lab. I try to get a recent picture, but you know, one of the challenges of doing academic research is you only have people in the lab for 3 to 5 years if you're doing things right, because they get trained and they move on and they go do it themselves. So I just want to highlight our MD PhD student from Florida Atlantic University and Scripps combined program, and our graduate student Christina Johnston, as well as to point out that our funding is from the National Institute on Health, including a fellowship to Agnes Acevedo-Cannibal, and to also acknowledge a foundation funding from, or a gift from Miss Florence Dembling from the Dembling Endowment Fund to our laboratory. She always really supported our work in the interface of depression and drug abuse. Thank you.
All right, well, thank you, Laura. As you heard, Laura's approach is really innovative. And you can see dealing with brain chemistry is quite complex. And I actually looked at this approach a number of years ago, probably over 30, but I wasn't as creative as Laura, so I went off on a different tangent. And I created this area what I call immunopharmacotherapy. And so basically biologics to try to treat substance abuse and disorders. I'm gonna basically frame this in a couple ways. I'm gonna introduce basically how these drug vaccines work. I know a lot of people have heard recently the COVID vaccines and what's being developed. So I want to kind of put this in a little bit perspective from this historical standpoint, and then go into how we develop our vaccines. Then the mechanism, how they work, reasons behind developing these vaccines, and opioids themselves, the PK/PD, so the pharmacokinetics, pharmacodynamics, how a drug reacts with the body, how the body interacts with the drug. And then I'll tell you about some of our vaccines in terms of the opioid abuse, heroin vaccine, a fentanyl vaccine, heroin contaminated fentanyl, and then carfentanil monoclonal antibodies. And then I'm going to do a little bit of a right turn. I'm going to move off from the opioids. I'm going to tell you what I think is a really cool piece of work we've been looking at, and it's a real problem today, and it's kind of been overlooked. I'm going to talk about a nicotine degrading enzyme. Okay, with that said, vaccines have been around a long time. The Chinese originally employed these as virilation. So basically taking dried pustules and then scratching their skin with this. I think most people have heard of Edward Jenner in the cowpox, where he recognized that the milkmaids were not being infected. And so he took some of these dried pustules and injected this young boy, and he was infected and he lived. It's considered the father of vaccinology. Most people don't know Louis Pasteur was one of the originally founders of a laboratory vaccine against cholera, I think in different areas. Moving ahead into the 20th century, I want to take a look at 1926 in Glennie. Alum is an adjuvant that is still used today, probably 90% of all vaccines, and it's over 100 years old. And I think you'll see in the future with these COVID vaccines, they don't have an adjuvant in them now, but to improve them, you're going to see an adjuvant being added to them in the future. Down the road from us, Salk vaccine in the 50s, probably people don't know that Kaprowski was actually before him in 48. And actually the March of Dimes was started by, you can see on the right, Eddie Cantor saying, "Send your dimes to President Roosevelt," and millions of dimes came in. That's how the March of Dimes started. And then one individual was overlooked, was a man named Hillman. Hillman's probably saved more lives than anybody at any time in the world. He's created probably 7 to 8 vaccines. Some are still used today, measles, rubella, and mumps. And like I said, vaccines that he's developed have been very safe and very effective. So what about the design of our vaccines? Are they very different from the vaccines that are out there? Not so much, but there are some differences. First of all, drugs aren't immunogenic. So you can ingest all the drugs you want, you're not going to get an immune response. So what we have to do is use the wizardry of synthetic chemistry and try to position them such that the body recognizes them as foreign. So we do this by putting a linker and putting them on a carrier protein, this is known as a T-cell epitope. We then formulate this with an adjuvant, and then we go ahead and vaccinate. I'm showing you a rodent in this case. So we've done mice and rats, but we've also done non-human primates, monkeys, in developing our vaccines. So the hope is that using this overall approach, we develop antibodies. Our own body would develop antibodies against the drug. And then we're going to test this out in different behavioral models to see if the vaccines work. A little more complex in how this works from the immunological standpoint is these vaccines are taken up by what are known as dendritic cells. They're then displayed on the MHC2, see there, and we get what's known as T-cell activation. These T cells help stimulate what are known as B cells, and we get clonal expansion of these B cells, which eventually make the antibodies that are going to basically be used to sequester the drug. If you don't like that complexity, a very simple model of this is shown here. Basically, antibodies don't cross the blood-brain barrier. Antibodies will block the drug from crossing into the brain where, as Laura talked about, there are receptors for them. The other thing that these antibodies can do is they can create a gradient, much like a vacuum, and they can draw the drug out of the brain. So there's two ways in which this immune pharmacotherapy works. So some of the opioid vaccines we developed over the years include morphine, mitragynine, heroin, oxycodone, hydrocodone, and the fentanyl analogs. So everything you see in red is some of the vaccines we've developed. You know, I'd be remiss not to say that there are medications for this. And so, you know, why are we looking at this? So as Laura mentioned, there's substitution therapy and such things as methadone and buprenorphine. But we would counter that it continues the opioid use, perpetuates dependence. There are possibilities for adverse reactions. It's costly to maintain. There's risk of drug aversion to other things, and you get a high attrition rate in the first month. So Laura also talked about antagonists, so naltrexone. But a real problem with this is it makes a lot of people who have substance use disorders sick. It can increase fatal overdose and relapse, and there's long-term side effects. I think we all know psychotherapy and cognitive behavior, but there's a lot, really problems with this is low compliance rates. So I would counter why an opioid vaccine is needed is that it doesn't require continual daily compliance. We believe it will allow physical dependence to normalize. It should reduce the risk of overdose. It should avoid adverse side effects, less costly to maintain. We believe that you shouldn't have a chance to other opioids, and scalably global. I mean, vaccines are used typically, you see a lot of these in third world countries. They're some of the most inexpensive therapeutics that are out there. And really when I got involved in working in the opioid problem, it wasn't to be used in the US, it was actually to be used in third world countries where I saw a spike in HIV with injectable drug users, which were also using heroin, and so the spread there. So that was the original intent for this work. So I want to kind of frame this to COVID-19. Jamie mentioned something about the two coming together. I kind of looked at as this is the perfect storm. With opioid use disorders, you have a heightened risk of pulmonary infections. This is because compromised immunity. With COVID-19 is sequestering, people can't get adequate access to healthcare delivery, failure to rehabilitate strategies because we have social distancing is a problem. Another thing which people don't realize is that a lot of opioid users misinterpret COVID-19 symptoms with withdrawal, and they try to treat this by self-administering opioids. But some of the biggest problems is really pretty simple, is people are isolated, they're stressed out, and when this happens, to try to alleviate these negative feelings, they turn to drugs. It's pretty simple as that. So I like to frame the heroin use with Bayer was one of the first commercial sources, it was used for a specific relief of coughs. As you can imagine, it could be used for almost any cough. One thing that's a little bit of misnomer with heroin, heroin's really a prodrug from morphine. Heroin doesn't really have, as Laura talked about, these mu opioid receptor binding affinity. It's really used because it gets fast access to the brain. So I like to consider heroin as a prodrug, and 6-acetyl-morphine and morphine is the real drug. So in developing these vaccines, the question is, what do we want to develop our immune response to? Do we want to make it to heroin? Do we want to make it to 6-acetyl-morphine? Do we want to make it to morphine? So I kind of call this a moving target. And so instead of showing some chemical structures, I'm going to show you a sedation slide where we injected a mouse so it is vaccinated against heroin, and then we're going to treat another mouse with a low dose of heroin and is basically sedated in the corner. And hopefully you can see the differences between these two. And you can see the one mouse that was vaccinated is getting a boatload of heroin, 18 mg per kg, so almost 10 times more. You can see the sedated one's just sitting in the corner there, where the other one has 10-fold excess of heroin. It's happy as a little clam moving around, and actually see he wants to get out of the box. Okay, so hopefully you can see the power of the vaccine in terms of basically supplanting the heroin's addictive properties. So I want to move into, as also Laura mentioned, fentanyl. Fentanyl was introduced by Janssen Pharmaceuticals in the 60s. So the main fentanyl derivatives are used as therapeutics are sufentanil, alfentanil, fentanyl itself, and low fentanyl. Carfentanil you see in the corner is used by veterinarians, usually to take down things like bears or rhinoceros, large animals, and I'll get back to that in a minute. Typically, the fentanyl analogs are used as in terms of surgical anesthesia or transdermal patches. So a big issue with fentanyl we've seen come up over the years, this has been since the 80s, is trying to skirt the DEA. So there's many different fentanyl derivatives made in clandestine labs. So thing called acetyl fentanyl. This was something that cropped up in Rhode Island a few years ago that killed a lot of people. Alpha methylfentanyl is known as China White. This was in the 80s, came into California. As Laura also talked about, why are some of these things coming into view? So it's really based on the economics. So when heroin becomes less pure, fentanyl is put into the mixture. When heroin becomes more expensive, fentanyl comes into the mixture. We don't see that many fentanyl overdoses with heroin on the West Coast because most of the West Coast heroin is what's known as black tar, which is basically a shoe polish. East Coast, they don't use this much. You see more fentanyl cropping in with cocaine in terms of overdose, in terms of mixtures. We're also talked about the respiratory failure, which is involved with fentanyl. And fentanyl derivatives do bind very tightly to the mu opioid receptors. But another big reason why fentanyl causes respiratory failure so quickly is how quick it gets out of the blood into the brain. So you can see in the bottom, morphine is very slow, hours, where these fentanyl derivatives get into the brain very quickly. So that's why you need to get something in there to take care of it, is because of its quick access to the brain. I'm not going to show you the fentanyl vaccines we delivered. I'll just show you one slide which shows that we can get complete protection in terms of vaccinating animals with fentanyl vaccines against fentanyl doses shown here. So this is nothing to do with pain. This is percent survival. Another thing that we've looked at is what's known as heroin contaminated fentanyl, so adulterated with this. You can see there at the bottom where it says any dealer's worth of their salt are not going to have more than 15 to 20% fentanyl in the product, because it'll kill everyone. But I think people have to realize addicts try to find the best high possible. And so it's interesting, they'll go where people are dying because they think that's the best high possible. So this is kind of a busy slide, but what we do to measure if this heroin fentanyl vaccine works is we look at how much fentanyl heroin's in the blood and how much is in the brain. We want to stop it from getting in the brain. And so the combination vaccine blocks both drugs, whereas we use each individual vaccine, it doesn't stop the other one. To put this in perspective, the potency of fentanyl and carfentanil, you can see here. So that small little dot of carfentanil is the amount that is lethal to a human. So it's extremely potent. And so we look at another different type of strategy. So the strategy I talked about of using these vaccines, I talked about is what we call an active vaccine. So I want to talk a little bit about what we're using monoclonal antibodies or a passive vaccine. So making these antibodies outside the body, you want to call them synthetic antibodies, and then placing them back in. So these antibodies come in different manifolds, and so we can structure these manifolds such that we can control the half-life of how long the antibodies are on board the body, try to, in terms of combating the drug. So again, the PK and the PD of how these things interact. So this is a bit of a busy slide, but so what I'm trying to convey here is that with carfentanil, and Laura alluded to this, is what you can get is what's known as renarcotization or rebound effect. So naloxone is great at removing fentanyl from the receptors. It basically, in simple terms, it brushes it off the receptors. However, carfentanil, while being brushed off by naloxone, naloxone doesn't stay around a long time in the body. Carfentanil has a long half-life. So when naloxone wears off, carfentanil comes back in again. And so what we're showing here is that the antibodies are able to stay on board longer periods of time where naloxone doesn't. And so when we get this renarcotization, the antibodies block this effect. And this can be seen a little bit easier in terms of respiratory depression, which Laura also talked about. You can see at the top panel with the antibody, we start to see respiratory volume coming back, where in the bottom, where you see in red, naloxone starts dropping off again, and basically the animal undergoes respiratory depression again. So as Jamie alluded to, we worked on a number of drugs. I've worked on these vaccines for almost 30 years. We've worked out initially with cocaine, as I talked about in the left corner, opioids. We work on methamphetamine, and I want to talk to you a little bit about some nicotine work that we've done, just to finish up. So I'm not going to go into all the reasons why nicotine is so addictive, but it's mainly due to affecting the receptors, which Laura again alluded to. And nicotine is probably as addictive or more addictive than heroin. And there's a number of papers you can read on this. So nicotine is a very difficult substance to try to get off of. Now, the other thing that people don't realize is that nicotine is extremely toxic. Nicotine is as toxic as the synthetic fentanyl derivatives. And so why I'm bringing this up is because with vaping coming into play, and the colored or flavored liquid nicotine, which is out there, it's extremely deadly to children. We've seen a huge upswing in terms of overdose and deaths to children over the past few years. And there are no therapeutics for treating nicotine overdose, none. So I think this is a really important problem with the advent of this nicotine toxicity, and the vaping is not going to go away. And so to try to address this, we looked at vaccines for a while. We weren't very successful. And I came across this bacteria that actually grows on tobacco plants. So it uses nicotine as its sole source of carbon and nitrogen. You can see this complex pathway, but it breaks it down. It basically eats nicotine to survive. And so what we did was, I circled it blue, cloned out this first step, this enzyme we call NiCA2, and it degrades nicotine to a non-psychoactive or toxic product. And this looks complicated, but it's pretty simple. So we look at how much nicotine is in the blood and the brain. So we chronically administer nicotine over 5 days, and we give the enzyme, and you can see in red where the enzyme completely obliterates nicotine. There's no nicotine can be found in either the blood or the brain. And so this is important actually for behavioral work. This is work done by Olivier George when he was here at Scripps. He's now at UCSD. And so we looked at withdrawal symptoms. We looked at pain threshold. We also looked at irritability. And basically, as you might imagine, the animals don't see nicotine at all. And so we don't see any of the effects. So I just want to kind of summarize a little bit what I've tried to talk through quickly today is that we believe we can generate a robust immune response that can defeat the opioid high. I didn't talk about this, but we can make these vaccines very specific, so we don't have to, so we can avoid opioids that can be used clinically effective in the medicinal setting, so we can make them very specific. We believe the immune response duration can be ideal. As I showed you, we can prevent overdose. We're working on these vaccines to combat multiple opioids in one combination. I talked to you a little bit about these monoclonal antibodies. I didn't talk to you about the area where these are hopefully going to be used possibly as terrorist threats. So carfentanil is considered a chemical warfare threat. And so our military is definitely afraid of this. And so we're looking to help them use this type of approach. And then finally, I talked to you a little bit about using an enzyme to treat nicotine addiction and possible overdose. And so I'm not going to go back through all the years of all the people that worked on this, just the current group shown here. The support came from National Institute of Drug Abuse, the Skaggs Institute, ALSAM Foundation, the Shattuck Family Foundation. I just put up in the corner here, this is something that was in the San Diego Union. We got Halloween, I guess, maybe coming up, but a real issue we've seen recently over the past few years is that people are not vaccinating their kids. And so we've seen a real upsurge with terms of measles, which is deadly, and polio. And so I encourage people to please vaccinate their kids against these diseases. Vaccines are some of the safest therapies we have out there. And so that is all I have today. I just want to put one last slide up. This is Andrew Sue, which will be speaking at the next upcoming lecture series. And hopefully everyone will attend.
Terrific. Thanks a lot, Kim. Thanks, Laura. And please join us for the front row lecture with Andrew Sue. He's one of our computational biologists, and he's really got an interesting take on using crowdsourcing to do some very interesting studies in biology and medicine. Okay, so I want to do some Q&A. We've got a few questions from Louis Shanuck, sorry. And the question is the relationship of euphoria and to analgesia. So is euphoria assistive to the maintenance of analgesia with these compounds?
I can take a stab at that. I mean, so euphoria or a sense of well-being or a good feeling, and can we ever separate that perhaps from the pain relieving properties, I think is the gist of the question. And it's not really clear. Certainly something that makes you feel better is going to have some benefit, right? Like one of the approaches you give your child who has a stomachache, tell them go watch TV, and suddenly they're not complaining because they're feeling better and they're distracted. Mm-hmm. And again, it is one of the hardest things to beat in a clinical trial is placebo. So I think for some types of pain, it may be part of the treatment, but pain is very diverse and very different. And so this answer is something that we'd like to know, but we don't know the answer entirely.
So here's a question I think both of you could weigh in on. So the opioids need to enter the brain in order to give pain relief, but the antibodies prevent it from going to the brain. So if you're doing that to, let's say, to treat addiction, would you be compromised in your ability to get pain relief by the antibodies?
Yeah, it depending on the vaccine and how it's comprised against whatever drug, sure. But like I said, we can we try to make the vaccines very specific, so we can so like the heroin vaccine won't, oxy or hydrochloride, we're using that. Other things, naloxone, naltrexone, things like that, they won't cross over to that. So vaccines can be made very specific for their use.
Okay, so here's a question for maybe for both of you. So the studies that you both described are, it's preclinical research. You're trying to understand the mechanisms and come up with new compounds or new treatments. So how long would it be till you could actually have your compound or treatment into patients? And what's the prospects of getting pharmaceutical companies interested in supporting this work and bringing it to market? So you can both have a different story to tell. So Laura.
Okay. Yeah, so the one I used a lot as a tool, 1718, couldn't be a drug. It hits the HERG channel, so it has a heart liability. But we know how to get around that, and we've worked on these compounds. We have a lot of support from the National Institute on Drug Abuse, who have looked at our compounds and looked for selectivity. They've helped us with that information about the HERG channel and so forth. So we work towards this. As far as the market right now, it's hard to convince investors to try to make another opioid, given the fiasco that we're in the middle of. So we're hopeful that perhaps we can make an addition to what's understood maybe in substitution therapy, and use these compounds to understand the mechanism of how these receptors function in response to diverse ligands.
Mm-hmm. Kim, same question.
Yeah, I've discussed this over the last 30 years with every major drug company, and they do great work, but just not our area of interest. I think part of it is they don't want the liabilities. It's difficult to deal with addicts in terms of clinical trials. So I have tried a number of biotech companies also. There is a biotech company now which is licensed out my vaccines. It's called Cessation Therapeutics. And so they actually are trying to, moving forward with a monoclonal antibody. They're going to manufacture. This is one against carfentanil, fentanyl, many of the fentanyl derivatives. And so that is moving forward right now. So that's hopefully, this is coming from private funding, mainly Mark Pearson, who's on our board of directors and started the Pearson Foundation.
Right.
So I'm hopeful in the next 16 months or so, it should be in man.
Yeah. Maybe let's ask a more sort of personal question. So Laura, when you went to graduate school, how did you decide that you wanted to become, a pharmacologist? And how did you find your path to where you are?
Yeah, so I grew up in Appalachia, and I went to college at Virginia Tech. And at Virginia Tech, I had a major in biochemistry and chemistry, and I did undergraduate research in both fields. So a couple summers I did it in biochemistry, and I did a bit in chemistry as well. And it's interesting because I still am in that area, and I still followed some of that. And I also had a course in college called Nutrition in the Brain, and I really enjoyed that. And I also had a drug discovery kind of drug synthesis class that was talking more about drugs of abuse. And both of those were really fascinating to me. So I say it's really from my college experience and having that opportunity to do research as a student in a lab.
Mm-hmm. Mm-hmm.
Yeah.
Right. So Kim, you started out life as a chemist. So how did you come to this journey of working on vaccines for addiction?
Yeah, I was, as you said, working with Richard Lerner. We were doing catalytic antibodies. I actually worked with George Koob early on trying to develop agonist and antagonist for cocaine, and I failed miserably.
George is the director of National Institute for Drug Abuse, right?
National Institute of
Alcohol.
And yeah, I failed miserably. And since I was working on antibodies, I thought, why not, let's see if we can make an immune response against these drugs. And so I told George this, and George, a lot of the molecules I was developing didn't make the animals feel very good. And so George wasn't too keen on giving them another compound. So I told George this new idea, and he didn't really understand what I was doing. He said, is this gonna make my animals healthy? Absolutely. I said, okay. And so we were lucky actually we looked at cocaine first. This was back in, I think, 91 or 92. So.
Right. Yeah, so. Terrific. Everyone has their own story for how they got here. So it's pretty interesting. I think we're kind of at 5 after the hour, so there's a lot more questions. I wish we could get to them all. We can. We're going to ask our speakers to reply to them. We've recorded all of the chats, and we can get some answers out to you in private by email. So I'd just like to leave you with a couple thoughts. Both of the speakers today did a terrific job, and there's really a common theme, and that is using chemistry as a tool to understand biology with the hope of affecting major changes in medicine. And that's what Scripps Research is all about. We have a tremendous roster of faculty working on all kinds of problems, and we're really happy that you could join us this afternoon and hear about two really novel approaches to combating opioid addiction. So again, Jamie Williamson, I'm Executive Vice President here, and I'm pleased to welcome you to the front row lecture series, and thanks for listening, and we'll see you next time. Okay, be well.